Just a quick reminder, what we talked about last class. We talked about the eye and the refraction of light in the eye. To make sure that we are all on the same page, when the light comes into the eye, what are the two refractory structures? What are the two structures in the eye that actually change the direction of light rays? Cornea and lens. Which structure is more refractory? Cornea. But which one we can regulate? The lens. Which muscles regulate the lens? Shape of the lens. It's the circular muscles. They have little tiny ligaments attached to them. Look in your records, it's all there. Which muscles regulate the lens shape? Ciliary muscles. When we want to look at something close to us, what happens to the muscles? Do they contract or relax? When we read, ciliary muscles, do they contract or relax? Contract. When they contract, what happens to the lens? They bulge. What happens to the ligaments? They loose. Right? Ciliary zonules, the ciliary ligaments are loose because when muscle contracts, it shortens. Right? When we want to look at something far, what happens to the ciliary muscles? They relax. What happens to the lens? They flatten because ciliary zonules become tense. During this accommodation for the close view and far view, what happens to the pupil? When we look at something close, pupil is constricted, and when we look at something far, it is dilated. The last but not least, far vision is under control of which branch of ANS? Sympathetic, and the close vision is under control of parasympathetic. Would anyone dare to tell me which nerve, parasympathetic, sends the signals for reading? Optic is entirely sensory, and we are dealing with at least three muscles that work all together: ciliary, sphincter pupillae, and dilator pupillae. Now, parasympathetic response: constriction of the pupil, sphincter pupillae, but which nerve? Oculomotor, very good. Is that clear? Pathology, like really quick review. When somebody has nearsightedness, the eye anatomically is too long. In farsightedness, it's too short. In nearsightedness, we use what type of lens? Concave. In farsightedness, convex. Okay? And you can kind of figure it out because concave lens, they move the focal point far, while convex lens move the focal point closer. So if the eyes are short, the eyeball is short, you need to move the focal point closer, so you got to use convex lens. If the eyeball is too long, you need to move the focal point farther, you have to use the concave lens. Do I make sense right here? Good. Now let's talk about phototransduction. Remember, I told you when we were talking about neurons and synaptic transmission and excitatory and inhibitory postsynaptic potentials, that this knowledge will be invaluable all the way, further in this course, in AP1 and in AP2. I told you that, right? Remember, I told you that. Remember my words. Okay, and don't tell me that I didn't tell you that. Here, in this chapter, when we're going to talk about transduction of the signal, and we'll talk what transduction is, this is what we essentially going to come down to. Transmission of the signal between three neurons. Okay? Now, what is transduction? We mentioned it once or twice. I want to see if you remember what transduction is. Signal transduction. Okay, light. What is that? How can you define the light in physical terms? Electromagnetic wave. Yes, it's a wave, electromagnetic wave. Wave and particle at the same time, but let's not go that deep. Okay, what about sound? But it's absolutely a wave, but mechanical wave. What about taste? And smell? Chemicals. So we have chemicals, we have electromagnetic wave, and we have mechanical wave. What is the nature of the nervous signal? Nature, physical nature. What is that? A bunch of little guys screaming or just, kicking each other in the butt. What is the nature? Mechanical, magnetic, electrical. What is the nature? Electrical, right? When we sense something, when you hear sound, you hear my voice, right? But that sensory perception, in the end, is transformed into the electrical impulses in your nerves, in your cochlear nerve, in your auditory cortex. Does it make sense? That moment, that process, I would say, not moment, the process, when you change, when you transform the signal from the outside, chemical, physical, chemical, mechanical, electromagnetic signal, into the nervous signal, is called transduction. We can say light transduction, sound transduction, taste transduction. Does that make sense? Do you understand what transduction is? Because I'm going to be using that word, and I don't want, any ambiguity here. We got it? Huh? I will try, because I don't usually give like strict definition. I would say that transduction is the transformation of the outside signal in the electrical nervous signal. Does that make sense? Look, I'm not the type of person who, okay, a little digression. Some students complain, not only in my class, but in other classes too. Not only in I mean to other professors. You write your questions and the wording is different compared to your lectures. And our response usually is when you start working, people that talk to you, do they talk the same language that I used in my lectures? The answer is usually no. So you have to understand the point, the idea. So I hope you understand the idea of transduction. Okay? Now, what we're going to talk about is how light, photons of light or electromagnetic waves, pick either, are transformed into the signal in the nerves. Does that make sense? And first and foremost, we need to chat about photoreceptor cells. Cells that are actually transducing light into the nervous signal. Speaking of which, I wanted to mention one thing. Sorry, I will not even go back. I will draw it on the slide. Let's first of all recall the arrangement of cells in the neural layer of retina. So we have pigmented cells, right? We have a layer of pigmented cells. Does that make sense? Remember those? And then we have photoreceptor cells. Correct? And then we have bipolar cells. And then we have ganglion cells. And they go and form a neuron. Okay? Can you see that? So using this little picture, and using left and right, tell me, where does the light come from, left or right? Right. So you think that light comes from here. What else here? Here's the choroid, okay? And then there's sclera. So you tell me that light has to come through sclera and choroid, and then pigmented cells, and then photoreceptor cells. Where does the light come from? Left bottom. The other way around, right? So actually light is coming this way. If you would be an almighty creator, which, just for the joke, it's not a biological entity, but if you would be the almighty creator, what would you say about this arrangement of cells? Remember, the blue ones, are photoreceptor cells. These guys, they sense the light. What do you think? Is that a good? Yep. Perfect. That's a great definition. Actually, one of the best I've heard. It seems backwards. Does that make sense? It seems unnatural that light has to cross ganglion cell first, bipolar cell second, and only then reach photoreceptor cells. Well, guess what? Our vision, it's good, but it's by no means the best. And part of it is the anatomical arrangement of cells in the retina. Which arrangement would be better? Like best? Ganglion? Yep, that would be ideal, right? Well, guess what? There's an animal that has such arrangement. It's called squid. Squid. Fish doesn't match the squid in terms of the acuity of vision. Because in the muddy waters, with very little light, to hunt fast-moving prey, it has to have. So, arguably, but squids have best vision among all animals. Okay? Now, that actually, it's a little bit of a digression, but that actually tells another thing. I always tell students, nature is extremely bureaucratic, which means it is not always the most optimal solution that you're going to find in the physiological structures. What do I mean by that? This is not optimal solution. Is that sufficient for humans to survive? We hear, so it is sufficient. Does that make sense? If that wouldn't be, there would be no humans. So you have, what I'm trying to bring up is that when you run into some awkward physiological system that seems extremely inefficient or extremely resource inefficient, time ineffective, and you think that could be done better. That could be, but as long as it works, nobody is going to throw it out. As soon as the system becomes a burden, evolutionary burden, survival burden, fitness burden, that system will die out. But as long as it contributes to overall fitness of a population, it's going to stay there. Does that make sense? So let's go back to photoreceptor cells. They are neurons. And I can show you how they are neurons. Soma, dendrites, axons. Names are different though. This part, the part of the quote-unquote dendrite, is called outer fiber and contains the inner and the outer segment. The outer segment is submerged into the pigmented cells. Okay, so pigmented cells, they function, remember, to phagocytose the fragments, the debris that come from the photoreceptor cells. Photoreceptor cells are constantly pounded by light. And fragments of the membrane, they kind of fall off, they die, and blah, blah, blah. So all these fragments of the membrane are recycled in the pigmented cells. Does that make sense? The outer segments of rods and cones look like assembled by the number of discs. We can represent it sort of like this way. Okay, like many discs. What do you think? And those discs are essentially the folds of the membrane. Okay, so it's multiple folds of the membrane that form the outer segment. Now my question for you is, why? Why discs? Why folds of the membrane? By the end of this course, it should become a reflex. I'm serious. If somebody tells you folds, you tell, oh, increased surface area. Why do we need increased? So, here's the thing. That membrane contains photopigments. Photopigments are the molecules that are responsible for photoreception. Does that make sense? Follow me? The more area with photopigments we have, the more light the cell can receive. The more light the cell can absorb. Does that make sense? Okay? So discs increase area for photoreception. Rods, you know, that's rods. They separated. Discs separated. In the cones, it's pretty much like folds of the membrane. In terms of the flow of the signal, on the basic level, here's what's going on. Light is absorbed, received at the outer segment. Transduction happens. We're not talking yet about how it happens. It does happen. Transduction happens. The signal is transmitted to the bipolar cell, and from bipolar cell farther, you know, towards ganglion cell. Do that make sense? Okay. So essentially you can see that signal is received on the dendrites, somehow goes through soma, axon, and neurotransmitter is, and okay, something happens at the synapse. Does it make sense? So this this point between the photoreceptor cell and the bipolar cell is the synapse. Do you follow me? Believe me, so far it's all that you have learned that. Okay? You have learned that. Now, as I mentioned, photoreceptor cells are damaged fairly easily, so they are in the constant rebuilding mode. The fragments are like dissociated from the cell and, get recycled by the pigmented cells, and new fragments are added, and the recycled material may come back from pigmented cell to the photoreceptor cell and vice versa. So far, does that make sense? Sort of take-home message. 3 parts, all dendrite, soma, axon. Dendrite is inner and outer segments. Outer segments consist of discs. Discs contain photopigments that receive the light. Any questions? Go ahead. Photopigments is the most valuable part of the outer segment. They contain the chemical that is the most valuable part of photopigment. When light hits the cell, it contains not only visible light, there's UV, all kinds of stuff. Cell gets damaged. Fragments of the cell quite literally they depart from the cell, they fall off the cell. In order not to lose that valuable chemical, which we will talk about in like 10 minutes, in order not to lose that chemical, those pigment cells phagocytose that debris, take chemical and transport it back to photoreceptor cells, so it can be used again in the photopigment. Does it make sense? Another reason, of course, if there would be no phagocytosis, those cellular fragments, that cellular debris, would simply pile up like the garbage around the cells, and that would cause inflammation and all other kinds of horrible things. Does that make sense? Rods and cones. The comparison. We talked about it a little. Rods. Very sensitive. Often, one photon is enough, which means they can sense really dim light. Got it? Oh crap, cones. Cones need bright light. Right now, it's your cones that work. Rods rods do not participate in your visual perception right now. So essentially, rods are necessary for night vision and peripheral vision. Night vision is, I think, kind of obvious, right? Because in the night, the light is dim. And peripheral vision, the amount of light that enters from the periphery is just low. Cones, colored vision in the light. Rods have only one visual pigment, so rods are not able to distinguish colors. It's grayscale vision. So all they sense is the intensity of light. You got it? Cones, RGB system, red, green, blue. The same system that is used in the computer monitors. Okay. So when they overlap, we can see all those different colors. Does that make sense? So it's not like you have special type of cells for pink or orange or brown. You have just 3 types, but they spectrum of sensitivity overlap. Spectra of perception overlap. That makes sense? Okay? What makes rods even more sensitive comparing to cones? They have converging pathways, okay? And we use the analogy, I think we use the analogy of the chorus versus one singer. Okay, let me tell you something. I'm talking to you now. You all can hear me, I hope. Okay? Now imagine for a second that somebody walks into the bathroom. Can you hear me in the bathroom? Probably not. Seriously, but when you hear, can you hear me clear? I know that my spelling, my pronunciation are not perfect, but at least you can hear my words, right? You can distinguish them fairly easily. Does that make sense? Now imagine there's going to be 100 of me talking at the same time, with slightly different intonations, slightly different rhythm, and all that kind of stuff. Will you be able to hear me in the bathroom? There's a chance. There's a chance. I'm not saying, I mean, the walls are pretty thick, but I would be much louder. 100 of me would be much louder. The only problem, if any one of you ever went to the opera, one guy sings, it's hard to distinguish the words. Chorus sings, forget it. Unless you have a text in front of you, fat chance. It's indistinguishable because there are so many voices. Does that make sense? Same happens with rods. When you have 100 rods that converge on one ganglion, essentially converging on one fiber, one pathway that goes to the visual cortex, sensitivity is enormous, right? Even very dimmest light can be picked up, and those 100 rods together will scream sort of in one ganglion cell. Does that make sense? It will be a good signal, but the signal is going to be fuzzy. Do you understand what I'm trying to say? But on the other hand, cones, each and every cone cell in the fovea centralis has its own pathway to the visual cortex. Think about this. The size of the pixel, do you know what pixel is? You all know what the pixel is. New generation, you should know. The size of the pixel that is formed by rods can be 100 cells. The size of the pixel that is formed by cone is one cone. So resolution is much better in terms of cones. But to initiate that signal, to cross the threshold for action potential, cone must be activated very well, very strong. Does that make sense? So it kind of explains why you need bright light for perfect sharp vision, and dim light you can still see, but vision, the picture is not going to be that good. Do you understand that difference? Go ahead.
So in the case of like animals that are nocturnal, is it basically just that their rods aren't feeding to one cell, so then if there are more cells that the rods are feeding to or whatever, it makes their vision sharper?
So, say again the last part. Sorry.
Yeah, that was really weird, I guess. Kind of like how you said with the cones, there's only like one cell that's receiving it, or like there's not that many cones going to one cell. So in the cases of animals that have nocturnal vision, is their vision clearer because there's not so many rods?
I would say that they think about this. Let me switch for a second to this. Think about this. When we talk about nervous signal, we always talk about action potential. If this is the threshold, remember threshold for action potential, and you have one rod, and one rod will produce, and I'm hesitant to say, it's slightly different, but let's say you have graded potential. When you have 100 graded potentials like this, by adding up, they will make an action potential. Does that make sense? Like 100 adding up. If you have a cone, I'm getting to your question. Single light stimulation must produce action potential so you can see something. There is no alternative. There is no graded level. It must be action potentials right away. In nocturnal animals, most likely, this threshold so this is humans. This will be nocturnal animal. So even minimal stimulation will cause the upstroke. Does that make sense? Actually, fairly similar situation is in smell, humans compared to dogs. In humans, there are amacrine cells in the olfactory bulb. They are like a relay station on the way to the brain, and they have very high threshold for activation, which means there are now millions of smells in this room. Millions. Does that make sense? We are not getting overwhelmed by all these chemical molecules that fly around, because most of those molecules do not stimulate amacrine cells to the threshold level. The constant signal too small. Does that make sense? So same happens, I suppose, with nocturnal animals. Got it? So, what if somebody, well, okay, let's talk about photopigments first, and then we're going to talk about color blindness. Photopigments consist of two components. The protein component, which is called opsin. You can see on this picture, the protein component is represented in the form of, can you see this gray ribbon? That's the protein component. Gray ribbon. And the chemical, organic chemical, polyene chemical, this one, it is called retinol. It's essentially a modification, a derivative of vitamin A. The same vitamin A, like if you eat a lot of carrots, carrots have orange color because of beta-carotene. Beta-carotene is a precursor for retinoic acid, which is vitamin A. Retinoic acid, when it is reduced, becomes retinol. You don't have to know that chemistry. It's for my personal enjoyment. I love chemistry, okay? But the idea is that vitamin A is essential for vision. Does that make sense? Okay, so you have two components. Protein component, and the protein is, as you can see, transmembrane protein. Okay, so it's embedded in the membrane, and you have a chemical component. Got it? So you have four different, like vast majority of people, practically every person, has four different types of photopigments. Rods have their own photopigment, okay? And all three different types of cones have their own photopigments. Got it? So we have four different types of photopigments. We often call photopigments altogether rhodopsin. Rhodopsin belongs to, rod photopigment, and we're going to talk about light transduction in rods because it's just simpler. But the mechanism is universal for all different types of photoreceptor cells. So far, are you with me? Opsin, retinol, all that kind of stuff. So different opsins, different proteins are different for different photopigments. Opsin for rods is going to be one. Opsin for red cones, another. Green cones, another. Blue cones, another. Okay? And we talked about the overlap of spectra already. Combination of red and green gives you yellow. Okay? Does that make sense? Other, I don't know, red and blue will give you something like purple. When they are all stimulated equally, you see white. I worked in printing technology when I was a student. You know, like printing machines, printing on the packaging. One of the very common misperception, if you take all the ink that you can get and mix it all together, it's going to be white. No, it's not going to be white. It's going to be horrible. Like very unpleasantly green, like vomit. Okay. So we have 4 different types of photopigments. 4 different proteins. Proteins. What do they consist of? Which elementary building blocks, which molecules they consist of? Nope, amino acids. Amino acids. So it's a bunch of amino acids, a chain of amino acids. The sequence of the protein, the information about the protein buildup, where is this information stored? In the DNA. And generally, 1 protein, 1 gene. Got it? So in people with normal vision, all 4 genes are active. Does that make sense? In people with color blindness, these genes are some genes are either not expressed at all, so there's no protein produced, or they are expressed at the lower level. Make sense? This condition is more common in males. And general name anomaly is the lower level of pigment. Anopia is the lack of pigment. Okay? Does that make sense? So for instance, protanopia is the lack of red pigment right here. Protanomaly is the lower level of red pigment. You can see that people with a protanomaly see a little bit of red, just a shades. Does that make sense? So what you need to know about this condition is this prefixes. Prota refers to red, deutero, green, and tridan to blue. Is that clear? Am I clear? Understand the difference between anomaly and anopia. Anomaly is the low level, anopia is the absence. Does that make sense? So it creates all various, in my opinion, it's quite exciting spectrum of color blindness. So this, I love this picture. So the person with deuteranomaly, the person who doesn't see green as well, that's the perception. So you see, that's the green skittle. And that's the green skittle. It's still green. It's not as green as it can be. Does that make sense? Like protanopia, absolute lack of red perception. Pick any, I don't know, this red, it's just gray. Okay? Tritanopia, lack of blue perception. We don't have blue skittles, by the way, why? No, it's just a rhetorical question. Why don't we have blue skittles? I don't know. But it changes the perception of some of the colors too. And it's very interesting. I don't know if you've noticed it in your life. Since I was working in that goddamn printing facility, every time the facility, and I was ink specialist, so I was responsible for colors being right. So when they were printing new design, I adjusted the colors, and then I brought the printout to the shift supervisor and told him, dude, is that good? And he compared it to like previous printout and said, yep, that's matching. Does that make sense? One time, we have a printout, and I pick the new one, I pick the old one, I compare them, they match perfectly. In my eyes, just brilliant match. I bring it to the shift supervisor, he looks at them, nope, this is more yellow. So he and we had different perception of what I thought was the same color, but probably the ratio of pigments was slightly different, and he had totally different perception. For him, one is way more yellow. He said, no, 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 no match. Go and adjust, and we couldn't get agreed on it. And you may notice it quite often that people have slightly different perception of colors. And on a little tangent, perception of colors in the language depends on where you live. You may have all heard that Eskimos people have something like 20 names for white, which is snow, because they know so many different types of snow. There is an Aboriginal tribe in South America that live deep in the center of the continent. They don't have a word for one of the colors. They don't have a word for blue. They do not have blue in the environment. They have, it's mostly grayish because of the rain. And when it's blue, they have something different. It's like a combination of green and black and something, but they don't have a word for blue. Especially, they're not exposed to blue ocean. They don't have true blue color in the environment, which is pretty crazy. Okay, so that's pretty common. If you don't have something around you, you're not going to have a word for it. Does that make sense? So what if someone has two cones missing? Is that possible? Yes. It's called monochromacy. So imagine you have one road and one cone. Okay? And the worst of all is achromatopsia. It looks like you live in the 50s all the time. Everything is grayscale. Okay? Yes?
So monochromacy is just you only see one color? Like
Grayscale. Everything is grayscale. You have roads.
well then is there any real difference between the achromatopsia and monochromacy?
In monochromacy, you have a road and a cone. You may have...
As far as like vision goes.
Oh yeah. There's going to be a little bit of color. Say if you have blue cone, you're going to be able to perceive blue colors. Everything else will go grayscale shades, mostly. Does that make sense? And sometimes it creates very unfortunate situations. I used to know a gentleman who, his dream was, well, he was a very old gentleman, but he told me that story. Absolutely true. It was in Louisiana. We lived in the same apartment complex. He said his dream was to be a pilot. And he couldn't because he had color blindness and everything in the cockpit was color-coded. Too bad. That's sad. Yes. So you have color vision. I don't know what type of cone you are missing or have lower levels of. As this shows, you still have colored vision. It's just the ability to distinguish like all the colors is impaired. Does that make sense? So you may not be able to perceive as much red color as other people can. Does that make sense? So, for instance, if you would compare normal vision here and Deuter anomaly, the lower level of green cone, the difference isn't that big. It's subtle. Green perception, blue perception is actually kind of expanded. So what other people may see as purple or violet, I'm always mixed up about those names of those colors, you will see as blue. Does that make sense? I don't know which cone is affected. Does that make sense? Oh, they have this circles with numbers. Yeah, that's how they test color blindness. Now the question is how what happens on the molecular level when light gets to photoreceptors. Retinol, the product of oxidation of the vitamin A. Okay, you don't need to know that it's a product of oxidation of vitamin A. It's, again, chemistry. Retinol is the huge organic molecule, as you can see from, that picture that I put the arrow to. And it's in a certain conformation, which is called 11-cis retinol. 11-cis refers to conformation of one of the double bonds, if you're too curious to know. Okay? So 11-cis retinol is bound to opsin, and this binding happens in the dark. Okay? When complex of opsin and 11-cis retinol is exposed to the light, 11-cis retinol changes its conformation. In chemistry it is called isomerized into all trans retinol. Do you follow? When retinol is changed to all trans, it dissociates from opsin. So you see they are separated. And that initiates the signaling. So opsin, when all trans retinol is away from it, opsin changes its conformation. It changes its shape. Does that make sense? You can try and make sort of a personal representation of what's going on. Imagine that I am opsin, and this pencil is 11-cis retinol. Okay, and here I am, formed in the dark, bound to 11-cis retinol. When light is absorbed by me, when I absorb light, 11-cis changes to all trans and dissociates from me, and I change my conformation. Does that make sense? That sequence of events. So far, that's it. So far, that's all you need to understand right now. Is that clear? Opsin translocates, goes away from the membrane, okay, into the cell, where it binds 11-cis retinol again and returns back into the membrane. But this process of return, okay, is called can happen only in the darkness. Does that make sense? So right now, is there a lot of light? There's a lot of light. All this light, what does it do to 11-cis retinol? It changes it to all trans. What happens to complex between opsin and all trans? They are what? They separated, right? So when they are separated, can they sense light? (11-cis is gone. 11-cis, this molecule is what absorbs the light. If this molecule is gone, how can it absorb the light? It's all changed, They're all out, Does that make sense? This is the form. That's the form, the opsin with 11-cis retinol. This is the complex that can absorb light. If they all separated, they cannot. This is called bleaching. Right now, your rods are bleached completely. Your rods have no functional photopigment right now. Opsin and all transretinal are all separated. Does that make sense? Do you see what I'm trying to say? Look, again, me. 11 cis. I'm opsin. Okay, so we form happy rod opsin. We can absorb light. It's not so much me, it's this molecule that can absorb light. When it absorbs light, it changes to all trans and dissociates from me. Can I absorb light now? I cannot. Well, I also change conformation, but that's a different story. Okay? Now, imagine light is gone, it's all dark. Go back, I bind to 11 cis retinal and I'm already functioning, and I'm again functioning. Does that make sense? Now, if there is constantly light pounding, any of the attempts to make a complex will be immediately halted. Does that make sense? Because there will be no 11 cis retinal molecules. They will be constantly converted to trans. Does that make sense? Okay? We got it? Now let's see what's going on afterwards. So let's recall the events that we just discussed in the previous slide. So, light is absorbed by 11 cis retinol. Does that make sense? 11 cis retinol is a component of the molecule called rhodopsin, which consists of this 11 cis retinol and the protein component, which is the transmembrane protein. The cytoplasmic side of that protein, the part that is in the cytoplasm of the photoreceptor cell, is bound to a special protein called transducin. It is the class of proteins, it's called G protein. You've already seen those. Does anyone remember when? If not, then not. Remember, we talked about two different types of receptors on postsynaptic neurons. One is ionotropic, ion channels, and one is metabotropic. So this is the type of metabotropic receptor. What happens is, that's a slower one. Very good. That's a slower one. So when 11 cis retinol is converted to all trans and is separated from opsin, opsin changes the conformation. That changed conformation activates the G protein. So this G protein is activated. G protein is then moves, the smart word is translocates, to the enzyme called phosphodiesterase. Phosphodiesterase breaks down the molecule of cGMP, converting it to GMP. So far, I know it's complicated, but let's go over it one more time. Let me try to make a little picture out of it. Okay, so we have a membrane. And in that membrane we have opsin. And in that opsin we have 11 cis retinol. I apologize for eye bleeding pictures. What happens when the light is absorbed by this photopigment? What happens to the 11 cis opsin? it becomes all trans and disconnects. So if you don't mind, I just remove it. Okay? G protein that is bound to the opsin portion on the cytoplasmic side becomes activated and moves toward the enzyme that is called phosphodiesterase. Does that make sense? So far, do you understand these steps? And it activates phosphodiesterase. So phosphodiesterase starts to break down the molecule called cyclic GMP to GMP. What's going to happen to the levels of cyclic GMP? They're going to decrease. Keep this thought. Turns out, cyclic GMP is the ligand for the cation channel, and it keeps cation channel open. If levels of cyclic GMP decrease, what happens to the cation channel? It will close. It will close. Does that make sense? If it will close, what's going to happen to the flow of sodium and calcium into the cell? It will stop. When flow of positive ions into the cell stops, positively charged ions do not go into the cell anymore. What's going to happen to the membrane potential of the cell? It's going to be more negative, which is called hyperpolarization. Very good. Does that make sense? So, skipping all the steps, light, how does it change the membrane potential of photoreceptor cell? It hyperpolarizes photoreceptor cell. Does that make sense? Okay? Do you want to go over these steps one more time without the slide, so you can tell me what's going on? What happens first? Let's try to make this picture again. So what happens first? Light. Huh? Light is absorbed. Let's not rush ahead of the train. Light is absorbed. Are we clear? When light is absorbed, what happens to 11-cis? You know what's cool? This is all get recorded. It's like going to be a little cartoon. Okay? So 11-cis retinol. What happens to 11-cis when light is absorbed? And dissociates from opsin. Okay? What happens to the G protein that is bound to opsin? Opsin changes the conformation, G protein gets activated. Activated G protein, what does it stimulate? You can say enzyme. I'm fine with that so far. Enzyme. You will learn the name later. Phosphodiesterase. Enzyme. Phosphodiesterase. Okay? So phosphodiesterase gets activated. What does it do? It breaks down some molecule, right? Again, I want you to focus on the concept, the conceptual part. So some molecule, some signaling molecule gets destroyed. It's cGMP gets destroyed. Okay? So cGMP is destroyed, and you have channels cGMP keeps these channels what? Open. So when cGMP is bound, channel is open. What goes through that channel? Sodium and calcium, right? So it's semi-selective channel. It allows sodium and calcium, but not potassium. Does that make sense? When cGMP is gone, what happens to the channel? It closes. So there is no flow of positively charged ions into the cell, and cell depolarizes. Does that make sense? Good? If you can sort of recount, that's sort of a take-home message from that picture. If you can recount the sequence of events, that is perfect. That's what you need. There are going to be several questions regarding that. Hyper. IROD, sorry about that, and great job. No. Well, we had a lecture in physical chemistry. The old dude, like 80 years old dude, who was deaf, like mortally deaf. He couldn't hear anything, and that's why he had enormously loud voice. And he lectured like all chalkboard, no PowerPoints, like nothing, just chalkboard. But he intentionally did 2-3 mistakes in each lecture to see if we can spot it. Yeah. Now, I'll be honest, there's no intention here. It wasn't my intention, and I appreciate you noticing it. Great, you pay attention. So indeed, cell hyperpolarizes. Hyperpolarization, in terms of, being like graded potential thing, what does that mean, hyperpolarization? Let's remind ourselves. So this is resting membrane potential, right? This is hyperpolarization, right? Membrane potential becomes more negative. Photoreceptor cells constantly produce neurotransmitters. Let it sink in. They constantly release neurotransmitters. When they become hyperpolarized, what happens to the neurotransmitter release? It stops. It stops. It stops. Okay? Does that make sense? Now, we're going to talk about the sequence of events between the photoreceptor cell, bipolar cell, and ganglion cell. And then, if it's not very clear, we could do another drawing on that white thing, but I would rather ask a couple of you to do some kind of, or 3 of you, to make a chain of photoreceptor, bipolar, and ganglion cell, and we will just show how it works. Okay? Does that make sense? So, photoreceptor cells and bipolar cells, they don't have action potentials in them. They generate excitatory postsynaptic and inhibitory postsynaptic potentials. Why? Action potential has to go distance. Think about this, you have ganglion cell, which axon extends all the way to where? Axons of ganglion cells, what do they form? The optic nerve. So it goes all the way to the brain. Those axons are pretty long. Does that make sense? So if you want to transmit information through that axon, you do need action potential. If all you need to do is to give signal from one cell to another, you just need graded excitatory postsynaptic or inhibitory postsynaptic potential. Does that make sense? Here's what happens. In the dark, to the left, in the dark, channels, those channels are open or closed in the dark. Open. Before we move on, everybody understands why in the dark cation channels, the membrane of photoreceptor cells, are open. Huh? No light, so it means PDE is not activated, there's no signaling, G protein signaling, there's no activation of phosphodiesterase, it means there is no breakdown of cGMP, so channels are open. So those ions, sodium and calcium, constantly flow into the cell. Does that make sense? What does calcium do in terms of the neurotransmitter release? It stimulates the release of neurotransmitter, and it does in case of the photoreceptor cell. So photoreceptor cell here, this synapse, constantly produces neurotransmitter, constantly releases neurotransmitter. So far, does that make sense? Here's the thing, that neurotransmitter is inhibitory one. Okay? It's glutamate, which is in this case inhibitory neurotransmitter. Does that make sense? Release of inhibitory neurotransmitter in that synapse inhibits, hyperpolarizes bipolar cell. Does that make sense? Okay, think about this. This is synapse, okay? This is neurotransmitter, and it's inhibitory. It will most likely close sodium channels, open chloride channels. So what's going to happen to this cell right here? The postsynaptic neuron, huh? Inhibited, yes. Inhibited, and great that you said more negative. More negative means hyperpolarized, right? Does that make sense? So bipolar cell is hyperpolarized. Are you with me? Are you on board?
So, what does the neurotransmitter do again in the synapse that makes it inhibitory?
It is inhibitory neurotransmitter.
And then what does that do?
It hyperpolarizes bipolar cell.
Because of the release of, what about the channels? You said like some of them are...
Chloride channels open. Bipolar cell becomes more negative. The membrane potential becomes more negative. It's by definition hyperpolarization. And hyperpolarization prevents, inhibits the release of neurotransmitter here. Because calcium channels are closed. That make sense? So that's the situation in the dark. Okay? What happens in the light? What happens to the ion channels in the photoreceptor cells? They close, right? Which prevents the flow of what? And sodium. What happens to the photoreceptor cell? It becomes hyperpolarized. Hyperpolarized cell, does it release neurotransmitter? Not anymore. What type of neurotransmitter does it not release? Say again? Inhibitory. So there is no inhibitory neurotransmitter at this synapse. So bipolar cell is not inhibited anymore. It is not hyperpolarized anymore. Its membrane potential increases. Does that make sense? And that increase in membrane potential opens calcium channels. When calcium channels are open, what happens to the calcium? Influxes into the cell. As it influxes in, what happens with neurotransmitter here? It is released. This neurotransmitter are excitatory. Release of neurotransmitter, what does it do to ganglion cell if it's excitatory? It excites it, yeah. Leading to the action potential or leading to depolarization of ganglion cell. Does that make sense to you? Okay. We're gonna pause it.